This is a demonstration of an aerobic bacteria test using the Dipslide method. Unscrew the cap and withdraw the cap and Dipslide from the vial. Be careful not to touch the agar-coated surface of the slide. Dip the slide into the water to be tested, covering the agar surface for a minimum of 3 seconds before removing. Allow excess fluid to drain from the slide. Remove the remaining drops by touching the end of the slide to sterile absorbent material. Screw the cap back on lightly and then back it off 1/2 turn. Incubate the vial in an upright position at 77 to 95 degrees Fahrenheit. 90 degrees Fahrenheit is generally good. For bacterial growth, the slide should be examined in the plastic vial 24 hours after incubation begins and after a total of 48 hours. For fungal growth, the slide should be examined after 48 hours and daily thereafter up to a total of 5 days. Bacteria grow as red or colorless colonies. To determine the number of bacteria, compare the colony density on the transparent slide with the colony density on the bacterial chart. Colorless colonies should be included in the comparisons. It is the number of colonies, not the size of colonies, that is important. Microbial counts in excess of 10 million may appear as a uniform pink or red layer. To obtain an accurate count on such samples, dilution is required. The brown side will detect the presence of yeast and/or fungi. Growth on this side may consist of pure yeast or fungi or a mixture of both. Growth and type should be recorded when first seen, but incubation should be continued for 4 to 5 days to obtain good evaluation of possible fungal contamination. Yeasts grow as smooth, round colonies, and fungi as fuzzy colonies. To estimate yeast levels, compare yeast growth on the fungal side to yeast colony density chart.